There's something faintly strange about the moment just before sleep. Not the darkness, not the quiet, something else, a kind of loosening, like the day, which felt so fixed and real 6 hours ago, is already becoming something slightly less solid. You probably didn't notice it happening. Most people don't. Welcome to the Sleepy Biologist, and if you're here, lying still, letting the day go blurry at the edges, then this space exists in part because of the people who've quietly kept it going. The channel members, the super thanks, the small gestures of support that don't get announced loudly anywhere. They're felt, and I'm glad you're here tonight. There's a library smell I associate with this. Not a good library, not the kind with high ceilings and reading lamps, but the kind with slightly warm air and fluorescent lights that hum just one tone above silence. I was reading about memory encoding in a room like that once, a long time ago, and I remember thinking the brain does more in an hour of sleep than I've done all day, and I wasn't wrong exactly, though I was missing the more interesting part of it. The interesting part is that the processing doesn't wait for sleep. It starts earlier than you'd expect. While you were sitting there, maybe at a screen, maybe eating something, maybe driving the same road home you always drive, neurons in your hippocampus were already beginning something. A kind of quiet sorting, not dramatic, not even detectable from the outside, but real and chemical, and happening on a timescale that makes right now feel like a slow concept. Here's what I mean. Every experience you had today, and there were more than you're consciously aware of, because your brain registered things your attention never bothered to visit. Each of those experiences left a mark. Not a permanent one yet, something more like a draft. A temporary electrically held trace that exists in a network of neurons that fire together in a particular pattern. The pattern is the memory. Or, it's closer to say the pattern is what will become the memory if the night goes the way it needs to. And that distinction matters. It's not that you experienced something and stored it. It's that you experienced something and began a process that, under the right conditions, will eventually make that experience retrievable. Those are different things. The second one is much stranger and much more fragile. Your hippocampus, tucked into each temporal lobe, one on either side, curved slightly like something that was folded once and never fully unfolded, spent today doing what it always does, taking in, tagging, holding things in a form of short-term biological custody. Is fussy about this, actually. Not everything gets tagged with the same urgency. Emotional weight changes the chemistry at the moment of encoding. Novelty changes it. Whether your attention was present changes it. The hippocampus is not a passive recorder. It is, in some way we still don't entirely understand, making decisions about what deserves to be held carefully and what can be held loosely. And those decisions are already made by the time you lie down. What shifts now, in this quiet, in the early minutes of letting the day go, is the relationship between the hippocampus and the rest of the brain. During waking hours, these structures are busy. They're receiving new input, updating constantly, handling the present moment. There's not much time or space for the slow work of consolidation. But when the external world goes quiet, when there's no more input arriving, no more decisions required, no more social navigation to do, something redistributes. The hippocampus begins replaying. Not randomly, it replays the day's traces in brief compressed bursts. Sequences that happened over hours are condensed into moments, and as they play, they are slowly, patiently transferred, not all at once, not cleanly, in toward the cortex, where they may eventually settle into something more durable. This is not instant. It won't finish tonight in any clean way. Some things from today will still be moving through this process weeks from now. That's not a glitch. That's how consolidation actually works. It's not a single event. It's an ongoing, layered process that keeps going quietly underneath everything else you do. There's something about that. I find it genuinely odd in a way that hasn't worn off, that your brain is revising today while you sleep. Not erasing it, revising. The version of today that exists in your memory tomorrow morning will not be a perfect copy of the version that existed at 4 p.m. It will be subtly altered, strengthened in some places, softened in others, contextualized in ways that hadn't happened yet while you were still living through it. And there's no version of today that doesn't go through this. Every day, without exception, gets handled this way. The ordinary ones, the ones you'll never think about again, the ones that felt important, all of them enter this same quiet process. All of them get worked on in the dark. The specific mechanisms, what moves and when and why certain memories survive while others fade, those are worth staying with. They are not simple. Even after decades of research, there are real gaps in what's understood. Not gaps that suggest we're wrong about the broad shape of it, but gaps in the fine grain of it. What's happening chemically? What determines which traces get replayed and how many times? Why sleep deprivation degrades some kinds of memory faster than others? Why a dream occasionally surfaces something you'd genuinely forgotten? Those questions don't have clean answers, and somehow I think that's the most honest thing about this whole subject, that the process running inside you right now, the one handling today, is still not fully mapped. Something is being done to the hours you just lived. You don't need to know exactly what. There is a point in the day, it shifts depending on the person, on the season, on how much the day asked of you, where something in the brain begins to feel slightly overloaded. Not dramatically, not the kind of overload that announces itself, more like a drawer that's been pushed too full to close properly. You may have felt it today without naming it, that particular flatness that settles in around late afternoon, where new information stops feeling interesting and starts feeling like weight. That feeling has a biological address. It lives, at least partly, in a structure most people have never thought about, tucked into the medial temporal lobe on both sides of the brain. One in each hemisphere, symmetrical small, carrying a disproportionate share of what it means to form a new memory at all, the hippocampus. I find myself drawn to this structure more than almost any other in the brain. Not because it's the most complex, it isn't particularly, but because it sits at such a strange intersection. It is both a temporary holding space and a relay point. It receives, it holds, it transfers, and it does all three of those things imperfectly, under biological constraints that most people never realize exist, while the rest of the brain continues demanding things of it without pause. What it does every night is, in some ways, a response to those constraints. A solution of sorts, though the word solution makes it sound more deliberate than biology tends to be. It's less a solution and more a pressure valve. A structural response to a problem the hippocampus cannot solve any other way. The problem is this: the hippocampus is not built for permanent storage. Its neurons are highly plastic, meaning they change their connection strengths relatively easily in response to experience, which is what makes them so good at encoding new information quickly. But that same plasticity is also a vulnerability. If the hippocampus were to hold everything it encoded at full strength, indefinitely, the connections would eventually saturate. New learning would begin interfering with old learning. The system would become, in a specific neurological sense, noisy, full of competing signals, none of which could be retrieved cleanly. This is not a theoretical concern. It is a real documented limitation in how hippocampal circuitry works. And the brain has been navigating it in some form for a very long time. The navigation happens at night. During waking hours, the hippocampus is in a mode that favors encoding. Acetylcholine, a neurotransmitter that rises during alertness, keeps it in a state suited to receiving new input. Connections form, traces are laid down. The day accumulates inside these circuits as a kind of temporary rapidly assembled record. But this mode is not compatible with the other thing the hippocampus needs to do, the transfer, the slow, careful relay of those temporary traces out toward the cortex, where they can be held in a more distributed, durable form, not as a single structure's burden, but as a pattern woven across many regions at once. That process requires a different neurochemical state, a quieter one where the hippocampus is not being asked to encode anything new and can instead turn its activity inward toward the records it has already made. Sleep provides that state. What happens during slow-wave sleep, the deep quiet phases that tend to dominate the earlier part of the night, is that the hippocampus begins replaying the day's traces, not as conscious experience, not as anything you would recognize as memory retrieval, but as brief, compressed reactivations of the same neural patterns that fired during the original experience. A conversation from this morning, a route you drove, the layout of a room you entered for the first time. These are not replayed in full, they are replayed in outline, the essential structure of the pattern, repeated sometimes dozens of times across the night, each repetition sending a faint signal toward the cortical regions that will eventually hold a more permanent version. The cortex, for its part, is not passive in this. It responds. Cortical neurons that receive these signals begin slowly to change, their connections strengthening in ways that reflect the incoming information. Over many nights, over many repetitions, the memory shifts its center of gravity. It becomes less dependent on the hippocampus, more distributed, more stable, more yours in some sense, though I'm not entirely sure that's the right way to put it. What stands out most to me, the part I keep returning to when I think about this, is the sheer patience of the process. There's no urgency in it. The hippocampus does not rush. The cortex does not demand. The transfer happens at biological speed, which is to say slowly, and in small increments, and across a time frame that human experience has no real feel for. A memory you form today may not be fully cortical for weeks. Some researchers think certain kinds of memories take months. The hippocampus holds them in the meantime, not passively but actively, repeatedly reactivating them, nudging the cortex, waiting for the slow structural changes to accumulate into something durable. And it does this every night, without instruction, without variation in its commitment to the task. There are sharp-wave ripples to brief high-frequency bursts of electrical activity in the hippocampus that appear to coordinate these replays. They last roughly 1/10 of a second each. During slow-wave sleep, they occur in coordination with slower oscillations in the cortex, the two rhythms nesting inside each other in a way that seems to create windows of communication between the structures. The hippocampus speaks during the ripple. The cortex listens during the slow wave. It is not a metaphor exactly. The timing really does work that way, in a measurable, consistent pattern that appears across many species, not just humans, which suggests it is old, which suggests it works. There are still things about this that aren't resolved, how the hippocampus selects which memories to replay preferentially, whether emotional content influences replay frequency, how the system handles memories that were only partially encoded, the ones where attention drifted, where something interrupted the moment before it could settle properly. Those questions are genuinely open, and I find that more interesting than unsettling. The idea that this structure, doing the quiet work inside you right now, is still not fully understood by the people who have spent careers trying to understand it. The hippocampus has a problem. It solves it the only way it can. And somewhere in the low electrical hum of slow-wave sleep, the solving has already begun. Not all stillness is the same. You know this already, even if you've never put it into words. The stillness of sitting quietly in a room is different from the stillness of concentration. The stillness just before sleep is different again, heavier, with a particular quality of loosening at the edges of thought. And sleep itself, which looks from the outside like one continuous quiet thing, is not that at all. I was sitting earlier, on the sofa actually, the kind of sitting where you're not really doing anything, just existing in the room, and I found myself thinking about a dream I had last night. It was strange in the way that only dreams manage to be strange, which is to say it felt completely normal while it was happening. There was a building I've never been in, a corridor that kept being slightly longer than I expected, someone I recognised but couldn't name standing at the end of it doing something I couldn't quite see. I woke up and the whole thing was already dissolving. Within a minute I had the corridor and the feeling, but not the person. Within 5 minutes I had just the feeling. By the time I made coffee, even that was mostly gone. I keep thinking about that corridor though, the way it stretched. Dreams do that. They borrow architecture from somewhere real and then quietly break the rules of it, and your sleeping brain doesn't object at all. But here's what's actually interesting about that. Not the corridor, the dissolving. The reason dreams vanish so quickly after waking isn't random, it's chemical, and it connects directly to what sleep stages are actually doing to your memories while the night moves through its phases. Sleep is not one state, it is several, cycling through the night in a rough pattern, though rough is the right word, because the pattern shifts and stretches depending on how tired you are, how much you drank, whether something woke you briefly at 3 a.m. without you fully registering it. The broad shape of it goes like this. After you fall asleep, you move through lighter stages into slow-wave sleep, the deep, quiet phases where the body does its heaviest restoration work, and where, as the last chapter covered, the hippocampus is running its replays. This slow-wave sleep is concentrated in the earlier part of the night. The first 2 or 3 cycles carry more of it. Then, as the night progresses, something shifts. The cycles begin to carry more REM, rapid eye movement, sleep. The stage most associated with vivid dreaming. The stage where the brain, in certain measurable ways, looks less like a sleeping brain and more like a waking one. And this shift, the gradual tilting of the night from slow wave toward REM, is not incidental. It reflects 2 different kinds of memory work happening in sequence. Slow wave sleep, as mentioned, favours the transfer of declarative memories, the kind you can consciously recall, facts, events, the specific texture of experiences, what happened, where, in what order. The hippocampus to cortex relay runs most actively here. REM does something different. During REM, the hippocampus is less dominant. The activity shifts toward associative cortical regions, the areas that hold not just individual memories, but the connections between them. What RM seems to be doing, at least in part, is integrating. Taking the newly transferred material and finding where it fits within the larger network of what you already know, not just storing, relating. There's a neurotransmitter called norepinephrine. It rises during stress, during alertness, during moments that need sharp attention. During REM sleep, it drops to nearly 0. This is one of the more specific and consistent findings in sleep research, and it matters because norepinephrine, at normal waking levels, appears to suppress certain kinds of associative thinking. When it disappears, connections that would normally be filtered out become possible. The brain, freed from that particular chemical restraint, begins finding similarities and patterns across memories that might not have seemed related during waking hours. A thing you learned last week and a thing you experienced today can suddenly sit adjacent to each other in a way the waking brain might never have arranged. Some researchers think this is part of why sleep, and specifically REM sleep, appears to support creative problem solving. Not because the brain is doing something magical, because it's doing something it can only do when a specific chemical is absent. And this, circling back, this is why the dream dissolved. When you wake from REM, norepinephrine returns quickly. The chemical environment that allowed the dream's logic, its loose, associative, rule-bending quality, collapses. The corridor that kept stretching, the person you almost recognized, those existed in a brain running without its usual filtering. The moment the filter returns, the dream's internal coherence has no support structure. It doesn't fade because dreams are unimportant. It fades because the state that generated it no longer exists. What's worth sitting with, and this is the part I find quietly odd every time I return to it, is that you move through these stages without any awareness of the transitions. You don't feel the shift from slow wave to REM. You don't notice when the hippocampal replays begin or end. You don't experience the norepinephrine dropping. The whole architecture of the night runs without you, and by morning it has done several distinct jobs that require different brain states, different chemistry, different structures taking the lead, and you wake up with no memory of any of it happening. Just the faint residue of a corridor or a feeling or nothing at all. The night is already moving through its first cycle. Somewhere in the earlier hours, the slow, careful transfer work is running. Later, and you won't feel the shift, the chemistry will change, and a different kind of processing will take over. More associative, more lateral, less about what happened and more about what it might connect to. Both matter. Neither announces itself. And the dream you might have before morning, the one you'll remember briefly and then lose, will be a byproduct of that second phase. Not noise, not randomness. Something that could only happen in a brain temporarily freed from its own constraints. It happens sometimes without warning. You're lying in the dark, the day mostly released, and something surfaces, not a recent thing, not something you were thinking about, but a specific afternoon from years ago. A kitchen you no longer live in, a particular quality of light through a window you haven't looked through since. The sound of something ordinary happening in the background of a moment you never consciously decided to keep. And you lie there thinking, why that? Why now? The honest answer is that we don't fully know, which I find oddly comforting in a way that's hard to explain precisely. There's something about a process this intimate, this personal, a process happening inside your own brain, selecting from the private archive of your own life, still having genuine mystery in it, still resisting a clean account. But there are things we do understand, and they're strange enough to be worth staying with. The brain does not store memories the way a cabinet stores files. This comparison gets made often, and it's wrong in almost every interesting way. A file stays where you put it. A file doesn't change between the time you store it and the time you retrieve it. A file has no relationship with the other files around it unless you deliberately create one. Memory is none of those things. What gets stored is not the experience itself, but a pattern, a particular configuration of neural connections spread across multiple brain regions that can be partially reactivated to reconstruct something that resembles the original experience. The reconstruction is never identical to what happened. It is built fresh each time, using the stored pattern as a kind of rough instruction, and the building process is influenced by everything that has happened since. This is the part that changes things. The Tuesday from 3 years ago, the one that surfaced just now, unexpectedly, without invitation, has not been sitting untouched since it happened. Every time it has been retrieved, even briefly, even partially, it became slightly unstable. open to revision. The connections that constitute it have been strengthened in some places and weakened in others, shaped by subsequent experiences, by related memories that were encoded near it in time, by the emotional context of every moment it was reactivated. What you remember now is not the original Tuesday. It is the most recent reconstruction of Tuesday, which carries the residue of every previous reconstruction. And the brain does this continuous quiet revision, not out of carelessness, but because it is the same mechanism that allows memories to be updated with new information. The same plasticity that makes learning possible makes perfect preservation impossible. You cannot have both. The brain chose learning. Now, the question of why that specific Tuesday. Why not a Wednesday from the same month or a Friday from the year before? Memory retrieval during the night, during the periods of hippocampal replay that run through slow-wave sleep, is not random, but the selection criteria are not fully mapped. What's understood is that memories with stronger emotional encoding tend to be reactivated more frequently. Memories that share structural similarity with recent experiences appear to be pulled up alongside those recent experiences, as though the brain is running a quiet comparison, checking the new thing against the old one, looking for overlap. This is where the Tuesday becomes interesting. Something from today, something you may not have consciously noticed as a connection, likely shares enough neural overlap with that Tuesday to have triggered its reactivation. A similar quality of light, a related emotional register, a sensory detail that was present in both moments, encoded into both memory traces, linking them in a way that has nothing to do with narrative logic and everything to do with pattern similarity. The brain is not telling a story, it is running a search, and the search returns results based on criteria you didn't set and can't directly inspect. There's a structure involved in this, beyond the hippocampus, called the entorhinal cortex, which sits just adjacent to it and acts as a kind of routing layer between the hippocampus and the rest of the brain. Information coming in and going out passes through it. And there's evidence that the entorhinal cortex plays a role in organizing memories not just by time, but by context, by the situational and emotional framework surrounding an experience. So memories encoded in similar contexts get stored in ways that make them more likely to be co-retrieved. They cluster, not physically, but functionally. And when one is reactivated, the others nearby in that functional space are gently pulled toward activation as well. This is, and I keep turning this over because it seems important, this is why memories often arrive in clusters. Why thinking about one summer tends to pull up other memories from that same summer, even ones you hadn't thought about in years. Not because they happened at the same time exactly, but because they were encoded in a similar internal state and the brain filed them accordingly. The Tuesday might have come with others, a feeling that preceded an image that preceded a sound. The sound not quite arriving before sleep shifted and carried the whole sequence somewhere else. This is how the night handles the past, not in clean labelled retrieval, but in associative drifts, in patterns finding patterns, in old traces briefly illuminated by the same neurological torch that lit a new experience earlier today. It doesn't mean the Tuesday was important, it means it was similar. Similar enough to something current that the brain's overnight comparison pulled it forward, held it briefly in whatever dim form of activation constitutes a nighttime memory replay, and then let it go again. By morning it may be gone entirely, or it may sit just at the edge of awareness. Not quite a memory you can place, more like a feeling with a colour attached to it. The specific Tuesday already dissolving back into the larger undifferentiated past, slightly altered from having been retrieved, slightly more connected to today than it was yesterday. The brain does this without asking. It reaches back, not randomly, but according to a logic you don't have access to, pulling old things forward to measure them against new ones, updating both in the process, then releasing them back into the dark. And somewhere in that process, in that quiet, unhurried overnight comparison, something that happened 3 years ago becomes, in some small molecular way, part of how you understand today. You started this night carrying today. Without knowing it, you're also carrying Tuesday. You already know this, not from reading it anywhere, just from living. The argument you had 7 years ago that you can still replay almost word for word. The afternoon something went wrong and you remember exactly where you were standing, what the light looked like, what you were holding in your hand at the moment you found out. And then, the ordinary Tuesday. Gone. Barely a smear. The brain doesn't apologize for this discrepancy. It doesn't pretend to treat all experiences equally. It has a system, an old one, built long before language, long before the kind of life where most of your experiences are safe, for marking certain moments as worth keeping. Worth keeping carefully. Worth reinforcing across multiple nights until the trace is deep enough that it won't easily blur. The system involves a small almond-shaped structure sitting close to the hippocampus on each side, the amygdala. It's tempting to call the amygdala a fear center. Neuroscientists have been pushing back on that description for years because it's too narrow and too dramatic both at once. The amygdala is not a siren. It's more like, and this is where the literal explanation serves better than any comparison, it is a relevance detector. It responds to emotional significance across a wide range. Fear, yes, but also desire, surprise, grief, the particular tightness of something that matters. When an experience carries that quality, the amygdala activates, and when it activates, it does something to the hippocampus next door. The mechanism is chemical. During emotionally significant moments, the amygdala triggers the release of stress-related hormones, norepinephrine, cortisol, that directly modulate how the hippocampus encodes the experience happening right now. The connections being formed are strengthened at the moment of formation. The trace is laid down harder in a sense, with more synaptic reinforcement than a neutral experience would receive. This is not metaphor. The molecular machinery of the synapse, the proteins involved in strengthening connections, the process called long-term potentiation, runs differently under these hormonal conditions. The emotional weight is not just a feeling you have about the memory. It is physically present in the structure of the memory trace itself. I've always found the timing of this striking in a way that feels slightly uncomfortable the longer you sit with it. The reinforcement happens during encoding, at the moment the experience is occurring, which means the brain is not waiting to see whether something turns out to be important. It is making a real-time bet based on your emotional state in that moment about how durable this memory should be. And those bets are not always right. A moment of intense anxiety encodes with the same chemical force as a moment of genuine danger. The amygdala responds to the emotional signal, not to an objective assessment of actual importance. So the memory of a public embarrassment from adolescence can be encoded with roughly the same neurological weight as something that genuinely threatened your safety. Both get held carefully, both get replayed, both get reinforced across nights of sleep. There's something worth pausing on here. The preferential consolidation of emotional memories during sleep, particularly during REM when the amygdala is notably active, means that the night is not a neutral sorting process. It has a bias. It leans toward the emotionally significant. It returns again and again to the things that carried weight. For most of human history this was useful. The experiences worth encoding deeply were usually the dangerous ones, the ones where something could be learned that might prevent harm. But in a life where most of your stressors are chronic rather than acute, where the things that activate your amygdala are emails and social situations and quiet ongoing worries, rather than immediate physical threat, the system still runs the same way. It still marks those things, still reinforces them, still brings them back in the night for another pass. The ordinary Tuesday doesn't stand a chance, really. It arrived without emotional signal. The amygdala barely noticed it. The hippocampus encoded it loosely, without chemical reinforcement, without the overnight replay priority that emotionally tagged memories receive, and so it fades. Not because nothing happened, because nothing happened that the brain's relevance detector registered as worth the metabolic cost of deep storage. What changes things, and this is the part that researchers are still working through, still refining, is not just the presence of emotion, but the type, the intensity, and something harder to quantify, the degree to which the experience felt unresolved at the moment of encoding. Unresolved experiences seem to receive more replay, more overnight attention, as though the brain is returning to them not just to reinforce them, but to, and this is speculative, I want to be careful here, to find an ending that wasn't available during the experience itself. Whether it ever finds one is a different question. Right now, while this night deepens and the body settles further into stillness, your amygdala is quieter than it was this afternoon. The emotional charge of the day is dispersing. But the memories it marked, the ones it flagged hours ago as significant, are already in the queue, already scheduled, in whatever loose biological sense scheduling applies here, for the attention they were promised at the moment they were formed. The brain keeps its own commitments. The things that mattered today will be handled tonight, not resolved necessarily, not made sense of, just held more carefully than the rest. There is a chemical that almost disappears from your brain while you dream, not reduced, not suppressed to a manageable background level, nearly absent, dropped to concentrations so low they barely register in the measurements researchers use to track such things, which are already measuring something almost vanishingly small to begin with. The chemical is norepinephrine, and its absence, it turns out, is not incidental to dreaming. It may be closer to the point of the whole thing. To understand why that matters, it helps to know what norepinephrine does when it is present, which is most of the time, during most of your waking hours, running quietly in the background of alertness and attention and the general effort of being a conscious organism navigating a world with consequences. Norepinephrine, released primarily from a small cluster of neurons in the brainstem called the locus coeruleus, keeps the brain in a state of what you might call disciplined connectivity. It sharpens signal-to-noise ratios. It makes the brain better at following a logical line, at filtering out associations that aren't immediately relevant, at staying on the thing it's supposed to be staying on. It is, in a functional sense, the chemical of focus, not in the dramatic caffeinated way, but in the quieter way of a mind that knows what it's doing, and is doing it without too much interference. When norepinephrine falls, that discipline dissolves. The locus coeruleus, and this is the detail that stays with me when I think about this whole system, does not wind down gradually across the night. It has periods of near total silence. During REM sleep, the neurons in this small brainstem cluster essentially stop firing, not slowed, not reduced to a murmur, stopped, in a way that has almost no equivalent in waking neurochemistry outside of certain extreme states that are not remotely restful. The brain during REM is running without one of its primary regulatory chemicals, and what happens in that absence is not chaos exactly. It is something more specific than chaos, a particular kind of loosened connectivity, where associations that would normally be filtered before they fully formed are allowed to complete themselves, where a memory of a corridor can stretch without the brain objecting, where two things that share almost nothing except a faint emotional resemblance can appear in the same moment without the usual requirement that their connection make logical sense. This is not the brain malfunctioning, it is, and I want to be careful here, because the temptation is to make this sound more purposeful than biology tends to be. It is the brain operating under a different set of rules. Rules that appear to serve functions that the norepinephrine-rich waking state cannot easily provide. The integration of distantly related memories, the softening of the emotional charge around difficult experiences. There's evidence that REM sleep, over multiple nights, gradually reduces the physiological response to emotionally loaded memories even while preserving the memory itself, a process sometimes described as emotional memory consolidation, though that phrase doesn't quite capture the strange specificity of what's happening. The discovery of hidden patterns across experiences that were encoded days or weeks apart. None of these things require consciousness. None of them require you to be aware they're occurring. They require norepinephrine to be absent. That's the main condition. There are other neurochemical shifts during REM that matter too. Acetylcholine rises significantly, which drives the intense neural activation that makes REM look electrically similar to waking states. Serotonin, like norepinephrine, drops. The motor cortex receives active inhibitory signals that prevent most of the body from acting out the movements the dreaming brain is generating. A separate system running in parallel, ensuring that the physical looseness of REM doesn't become literal physical movement. The body is held still while the brain runs loose. There's something about that pairing, the stillness of the body and the strange, unfiltered activity of the mind, that feels like it should mean something, though I'm not sure exactly what. It might mean nothing beyond the practical requirement that dreaming animals not exhaust themselves or injure themselves during sleep. But it is a striking combination. The most associatively free state the brain ever enters, paired with the most complete physical restraint it ever experiences outside of paralysis. Dreams, emerging from this chemistry, have a logic that is entirely their own, internally consistent in the moment, baffling in retrospect. The corridor that stretched. The person who was two people at once. The building that was somehow also a place from childhood. These are not errors in the dream's narrative. They are the expected outputs of a system running without its usual filtering. A system in which pattern completion is happening faster than logical evaluation, in which the brain is finishing thoughts before it has checked whether they make sense. The checking mechanism is offline. The norepinephrine that would normally intercept an association and ask whether it's relevant, whether it holds up, whether it connects to anything real, that chemical is not there to ask, and so the association completes, and then another one completes. And the dream accumulates not as a story being told, but as a series of pattern completions running in sequence, each one triggering the next, none of them filtered for coherence until the moment you wake up and norepinephrine floods back and the whole structure collapses under its own sudden implausibility. What remains in the seconds after waking is usually just the most recent completion, the end of the last sequence. And even that is fragile, held in a neurochemical environment that is actively hostile to the kind of loose associativity that generated it. Within minutes, the trace is gone, not because it was unimportant, but because the brain that remembered it is no longer the brain that made it. The chemistry has changed. The dreaming brain and the waking brain are, and this is not metaphor, this is a reasonable description of the neurochemical reality, genuinely different operating states, with different connectivity, different filtering, different rules about what counts as a valid association. You wake into one state and lose access to what was generated in the other. Every night, the locus coeruleus goes quiet. Every night, norepinephrine drops toward nothing, and the brain, freed from its own discipline, does the associative, integrative, emotionally processing work that the waking state cannot do. And then the chemical returns, the filtering resumes, and the day begins again with a brain that has been, in ways not fully visible from the inside, quietly reorganized by the night. The dream was a byproduct. The chemistry was the point. It would be simpler if it were, honestly, and I say this having just spent the better part of an hour trying to hold all of this in my head at once. It would be a lot simpler if consolidation were just one clean process, finishing one clean job and then being done with it. One mechanism, one time frame, one structure doing one thing, but it isn't. And the biology doesn't apologize for that. There are at least two distinct forms of consolidation running in parallel, operating on different timescales, using different structures, serving related but meaningfully different purposes. Researchers sometimes call them synaptic consolidation and systems consolidation, which are accurate names and almost completely unintuitive ones. Synaptic consolidation happens fast. Within hours of an experience, the synapses involved in encoding it undergo molecular changes. Proteins are synthesized, receptor densities shift. The physical structure of the connection between neurons changes in ways that make the pattern more steer, more resistant to being overwritten by the next thing that comes along. This process is local. It happens at the synapse itself. It doesn't require sleep, strictly speaking, though sleep appears to support and extend it. It is also on its own not enough. I keep coming back to that, not enough, and I'll be honest, after going around and around with this material tonight, that phrase feels slightly personal. Like the brain and I are both sitting here, having done a lot of work and being told it's still not finished. Systems consolidation is the slower process, the one that takes weeks, months, possibly years for certain kinds of memory. This is the hippocampus to cortex transfer, the gradual redistribution of a memory from a single structure's temporary hold into a more distributed, durable cortical pattern. And it runs every night, not completing, running. The distinction matters more than it might seem. When someone talks about consolidating a memory, they are almost always referring to synaptic consolidation, the fast local process that stabilizes a trace within hours. But that stability is not the same as permanence. A synaptically consolidated memory is stable in the short term, but still dependent on the hippocampus for retrieval. It hasn't yet become the kind of memory that survives hippocampal damage. It hasn't yet spread its roots into the cortex deeply enough to stand on its own. That takes time that synaptic consolidation simply does not provide. There is a patient, referred to in neuroscience literature as HM, though his full name was Henry Molaison, and he died in 2008, whose case clarified something essential about systems consolidation that would have taken much longer to understand otherwise. He had his hippocampus surgically removed on both sides to treat severe epilepsy. After the surgery, he could no longer form new long-term memories. Each day reset, each conversation was new. But his memories from years before the surgery, the ones that had completed systems consolidation, that had fully transferred to cortical storage, those remained largely intact. The hippocampus was gone, the old memories survived, which told researchers something precise, that the hippocampus is necessary for consolidation, but not for the storage of fully consolidated memories, that there is a point, reached slowly over years, at which a memory no longer needs the structure that originally held it. And here's the thing that gets me sitting with this at this hour. The process has no clear end point. There's no signal the brain sends when a memory has finished consolidating. No moment where systems consolidation completes and the hippocampus releases its hold and the cortex takes full custody. It is gradual in a way that resists being marked. Some researchers think certain memories never fully complete the transfer, that the hippocampus retains some involvement indefinitely, not as primary storage but as a kind of index, a way of accessing the contextual details that cortical storage tends to lose over time. The cortex keeps the gist, the hippocampus keeps the specifics, both apparently for a very long time. Tonight is somewhere in the middle of all of this. The synaptic consolidation of today's experiences is already underway, probably began hours ago, before sleep, in the cellular machinery of the synapses that encoded what happened. The system's consolidation of older memories is running its slow, patient background process, as it has every night, moving things incrementally toward cortical permanence. Neither will finish tonight. Neither is trying to. Consolidation is not an event. It is a condition, an ongoing biological state that the brain maintains across sleep and waking both, shifting its emphasis depending on the phase of the night, the age of the memory, the depth of the original encoding. It runs without urgency. It runs without announcement. And it will still be running tomorrow night and the night after, quietly working on things you've already forgotten you experienced, which is either deeply reassuring or mildly exhausting to contemplate, depending on how long you've been thinking about it. Something is being let go tonight. Not dramatically, not with any sensation you'd notice, but somewhere in the quiet electrical activity of a sleeping brain. Certain traces are being weakened rather than strengthened, their connection gently loosened, their retrieval probability nudged downward. Their hold on the network made slightly less firm than it was this morning. This is not damage. This is the other half of the process. There's a tendency, understandable, probably universal, to think of memory consolidation as purely additive, as the brain collecting and keeping, sorting and storing, building up a more complete record of a life with each passing night. And consolidation does do that. But it does something else simultaneously, something that rarely gets mentioned in the same breath. Something that is just as deliberate and just as biological. It forgets, actively, selectively on purpose. The concept has a name in neuroscience. Synaptic homeostasis, and the hypothesis built around it, developed largely by Giulio Tononi and Chiara Corelli, proposes something that initially sounds almost counterintuitive. During waking hours, synaptic connections across the brain generally strengthen. Every experience, every piece of information, every moment of attention leaves behind slightly stronger connections than existed before it. The network, across the course of a day, accumulates synaptic weight in a way that is metabolically expensive and, if left unchecked, eventually unsustainable. According to this hypothesis, is partly a reset, a broad distributed downscaling of synaptic strength, not erasing specific memories, but reducing the overall weight of the network to a level from which it can begin accumulating again tomorrow. What survives this downscaling is what matters. The memories encoded most strongly, the ones reinforced by emotional weight, by repetition, by attention, by the amygdala's chemical marking, those retain their relative strength even after the general reduction. They lose some absolute synaptic weight, but so does everything else, and they lose proportionally less. The weakly encoded traces, the unremarkable Tuesday, the name of someone you met briefly, the contents of a conversation that carried no particular charge, those lose more. Their connections were already thin. The downscaling thins them further. Some will survive in degraded form, some will not survive at all. By morning they are simply gone. There is something almost clarifying about this, if you sit with it long enough. The brain is not trying to keep everything. It was never trying to keep everything. The forgetting is not a failure of the system. It is the system working correctly, making room, preserving signal by reducing noise, ensuring that tomorrow's encoding doesn't have to compete with the accumulated weight of every unremarkable moment that came before it. A brain that kept everything with equal fidelity would, in a fairly specific neurological sense, become unable to learn. The new would be indistinguishable from the old. The important would be buried under the trivial. The connections that matter would be lost in a network where everything connected to everything else with equal weight. Forgetting is what makes remembering possible. But the selectivity of it is worth pausing on, because the brain's criteria for what gets kept and what gets released are not always aligned with what you would consciously choose to keep. The system optimizes for biological relevance, emotional significance, survival utility, novelty, not for the things you might personally want to preserve. The conversation you wish you could remember word for word may have been encoded without sufficient emotional signal to survive the night's downscaling. The embarrassing moment you would give anything to forget may have been encoded with exactly the kind of amygdala reinforcement that makes it nearly impossible to lose. The brain keeps what it decides matters, and it decided before you had any say in it. There's a separate process worth mentioning here: active systems consolidation, running in parallel with the downscaling, which is itself selective in a different way. During slow-wave sleep, the hippocampal replays that transfer memories toward the cortex do not replay everything equally. There is prioritization happening, though the exact mechanism of that prioritization is still being worked out. Memories tagged as emotionally relevant get more replay time. Memories connected to unresolved problems, to incomplete experiences, to things the brain has flagged as still open, those appear to receive preferential treatment. The brain is not neutral about what it chooses to work on overnight. It has preferences, biases built into the architecture. What this means practically is that the version of today that exists in your memory a week from now will be a curated version. Not falsified, the broad shape of what happened will remain. The emotionally significant moments will remain. The things that connected to existing knowledge will remain. But the texture will be thinner. The peripheral details will be gone. The moments that carried no signal will have been quietly released. And you won't notice the gaps. This is the strangest part of it, maybe. The forgetting is invisible from the inside. You don't experience the loss of a weakly encoded memory because the memory was never strong enough to leave an absence when it goes. There's no hole where it was. There's just nothing where something briefly was. Tonight, while the body is still and the night is doing its quiet work, this is happening alongside everything else. The strengthening and the weakening both. The keeping and the releasing simultaneously. The brain building a more durable version of today, while dismantling the parts of today that didn't earn their place. By morning, the record will be incomplete. It was always going to be incomplete. That's not a flaw in the system. It is, in some way, that takes a moment to settle. Exactly the point. There is something happening in the spaces between your neurons right now, not inside the cells themselves, not in the electrical firing, not in the synaptic chemistry that the last several chapters have been quietly circling, but in the gaps, the interstitial spaces, the narrow fluid-filled channels that run between brain cells like the thin passages between buildings in an old city, unremarkable from above, essential from within. A fluid is moving through those channels right now. It has been moving since you fell asleep, and it will keep moving until you wake, and most people go their entire lives without knowing it exists. The glymphatic system, the name is a combining of glial and lymphatic, which hints at its nature without quite capturing it, was only described in its current form in 2012, which is recent enough that it still carries a faint quality of newness in the literature. The kind of newness where researchers are still finding edges and exceptions and details that don't quite fit the initial picture. Maike Nedergaard and her colleagues at the University of Rochester identified it in mice, and subsequent work confirmed its presence and basic function in humans. And what they found was, I keep returning to this because the timing of it is the most striking part, that the system is dramatically more active during sleep than during waking. Not slightly more active, dramatically. The channels through which the fluid moves, cerebrospinal fluid driven by the pulsing of blood vessels and the slow rhythms of respiration, appear to be physically wider during sleep. The cells that surround them, called astrocytes, seem to shrink slightly, opening the spaces between them, allowing the fluid to move more freely and in greater volume than the waking brain permits. The brain, it seems, has been waiting for you to lie down. What the fluid carries out as it moves is metabolic waste. The byproducts of neural activity, the molecular debris that accumulates during a day of thinking and perceiving and remembering and deciding. Among these byproducts is a protein called amyloid beta, which has received considerable research attention because of its association with Alzheimer's disease, where it accumulates in plaques between neurons in ways that correlate with cognitive decline. The glymphatic system clears it. During sleep, the flow of cerebrospinal fluid through these channels flushes amyloid beta and other waste products out of the brain's interstitial space, carrying them toward the lymphatic vessels at the brain's borders and eventually into the body's general circulation for disposal. This is the extended metaphor I want to sit with for a moment, and then let go of, because it only carries so far. The sleeping brain as a city at night, its streets finally empty enough for the cleaning crews to move through. Not a perfect comparison. The brain is not a city, the fluid is not a crew, but the timing is genuinely similar. The activity that fills the space during the day is also the activity that prevents the cleaning from happening efficiently. The two processes appear to need different conditions. The waking brain and the washing brain cannot fully coexist. The metaphor ends there. What remains is the biology, which is strange enough on its own. There is a relationship between this system and sleep architecture, specifically between slow-wave sleep and glymphatic flow, that is still being mapped but appears to be meaningful. The slow oscillations of deep sleep, the large electrical waves that sweep through the cortex in that descending phase of the night, seem to drive the pulsing of cerebrospinal fluid through the glymphatic channels. The rhythm of the wave and the rhythm of the fluid movement are coupled in a way that suggests the deep sleep phases are not just neurologically important for memory consolidation, but physically important for the maintenance of the brain's cellular environment. The same sleep that consolidates memories is also washing the space those memories live in. Both processes running simultaneously in the same quiet hours, using the same general conditions of stillness and reduced external demand. Sleep deprivation studies, and these are uncomfortable to read, in the way that studies with clear implications for daily modern life tend to be, show that even a single night of poor sleep results in measurably higher levels of amyloid beta in the brain the following day. The glymphatic system did not have the time or the conditions to complete its work. The waste accumulated, and while a single night's accumulation is not by itself catastrophic, the brain is not so fragile. The pattern across many nights, across years, is what the research finds concerning. Chronic sleep insufficiency and the long-term accumulation of metabolic waste in the brain's interstitial space appear to be connected in ways that are not yet fully causal, but are consistent enough to be worth taking seriously. I find myself thinking about this more than is probably useful. There is also something the glymphatic system does that is less about clearing waste and more about distribution. It appears to facilitate the movement of certain molecules, including lipids and glucose, through the brain's interstitial space, acting as a kind of slow circulation system for the brain's own internal environment. The brain is not just being cleaned during sleep, it is being supplied, maintained. Its chemical environment is being adjusted and balanced in ways that the busy, waste-generating waking state makes difficult. The brain during sleep is, in this very physical sense, being cared for. Not metaphorically. The fluid is real, the channels are real, the molecular clearance is measurable and has been measured. Right now, while this chapter settles and the night deepens further, the cerebrospinal fluid is moving. The channels are open in the particular way that sleep allows. The astrocytes have made their quiet accommodation. The debris of today, the molecular cost of everything your brain did between waking and sleeping, every thought and perception and retrieval and decision is being carried toward the edges, toward the lymphatic borders, toward the body's long route of disposal. By morning, the interstitial space will be cleaner than it is right now, not perfect, not entirely cleared. The system is not that efficient and the night is not that long, but cleaner, quieter at the molecular level, more prepared for tomorrow's accumulation than it was when you closed your eyes. The fluid moves whether you know about it or not. It has always moved. The amygdala does not sleep the way the rest of the brain sleeps. During REM, the phase where most of the brain's activity shifts inward, where the filtering chemicals drop and the associative connections run loose, the amygdala remains notably, measurably active. Not in the sharp reactive way it fires during a frightening waking moment. Something slower than that, more sustained. Like a structure that has been handed something difficult and is sitting with it rather than reacting to it. This is not incidental. Fear memories, and by this I mean memories encoded under conditions of genuine threat, or perceived threat, which the brain handles identically because the amygdala cannot easily distinguish between the two. These memories enter the consolidation process carrying more chemical reinforcement than almost any other kind of experience. The encoding is deep. The synaptic weight is heavy from the moment of formation. The hippocampus, flooded with stress hormones at the moment of the original experience, lays down the trace with a specificity and a durability that ordinary experiences rarely receive. This is old biology, older than language, older than the kind of threats most people living now will ever actually encounter. The system was built for a world where remembering the location of a predator, the sound that preceded an attack, the smell of something dangerous, where remembering those things with absolute fidelity could be the difference between surviving and not. The depth of fear memory encoding is not a flaw. It is the system performing exactly as it was designed to perform. The problem is that the design is very old and the world has changed considerably. During REM sleep, the amygdala's sustained activity appears to serve a specific function in relation to these memories, one that researchers have been working to understand more precisely for several decades, with results that are interesting and somewhat incomplete in equal measure. The leading hypothesis, supported by a reasonable body of evidence, is that REM sleep facilitates a process sometimes called emotional memory consolidation, which is, I think, a slightly misleading name, because it implies the emotion is being preserved along with the memory, when what the evidence more clearly suggests is something closer to the opposite. REM sleep, over multiple nights, appears to reduce the physiological emotional response to a memory while leaving the factual content of the memory intact. The memory stays. The charge around it diminishes. This is worth sitting with, because it is genuinely strange when you look at it directly. The brain is not erasing the fear. It is not suppressing it, not filing it somewhere inaccessible, not resolving it in any narrative sense. The event that caused the fear remains in memory with its basic structure intact. What changes gradually across nights of REM sleep that keep returning to the memory's trace, that keep reactivating it in the low norepinephrine environment where emotional associations can be gently loosened, is the visceral intensity of the response when the memory is retrieved. You remember what happened, but the body's reaction to remembering it becomes slowly less acute. The cortisol spike softens, the heart rate change diminishes, the sense of immediate threat that the memory carries begins to detach incrementally from the factual content of the experience. This process takes time. Multiple nights, sometimes many nights, sometimes, for memories encoded under extreme conditions, a very long time indeed. There is a specific neurotransmitter involved in this that I find myself returning to. Norepinephrine, the same chemical whose absence during REM creates the conditions for dreaming, is also the chemical most associated with the acute stress response. Its near total disappearance during REM sleep creates, among other things, a neurochemical environment in which fear memories can be reactivated without the full physiological stress response that accompanied their original encoding. The memory plays back in some form in a brain that is not currently experiencing the stress hormones that were present when the memory was made. And this mismatch of reason between the memory's emotional content and the brain's current chemical state may be precisely what allows the emotional charge to be gradually revised downward. The brain revisits the memory without the body's alarm system fully engaged. Over many repetitions across many nights, the association between the memory content and the acute fear response appears to weaken. When this process is disrupted, when REM sleep is fragmented or shortened, when the normal cycling of the night is interrupted, the emotional processing of fear memories appears to stall. The memory remains, the factual content is preserved, but the physiological charge around it does not diminish in the usual way. The memory stays sharp in a specific sense, not sharper in detail but sharper in felt intensity. The body continues to respond to its retrieval as though the threat was still current. This is, as far as researchers can determine, part of what distinguishes normal fear memory processing from the kind of persistent intrusive fear memory that characterizes post-traumatic stress. Not simply the severity of the original experience, but what happens to the memory during subsequent sleep. Whether REM sleep is able to do its slow work of emotional revision, or whether something, the severity of the encoding, the disruption of sleep itself, other factors still being investigated, prevents that work from completing. The biology of trauma is not separate from the biology of sleep. They are, in a very specific mechanistic sense, the same story. Most fear memories, though, are ordinary ones. The near miss on a road, the moment something went suddenly wrong, the particular quality of dread that preceded bad news. These memories were encoded deeply, reinforced heavily, and they will receive preferential treatment during tonight's REM phases, the amygdala returning to them, the norepinephrine-depleted brain reactivating them in conditions that allow, slowly, for the charge to soften. Not tonight necessarily, maybe not for several nights, but the process is running. The amygdala is handling something right now that it was handed weeks or months or years ago and has been quietly working on ever since, in the particular way that only the sleeping brain's chemistry allows, not resolving it, not erasing it, just gently, patiently loosening what was wound too tight at the moment of formation. The fear stays. The body's relationship to it changes. That distinction, small as it sounds, is, I think, one of the more quietly important things sleep does. Not the most visible, not the most discussed, but running underneath everything else, every night, in the slow, sustained activity of a structure that does not fully rest, even when the rest of the brain has gone quiet. The amygdala holds things carefully, and in holding them across enough nights changes them. I have a slightly complicated relationship with this particular piece of biology, not because it's difficult to understand. It's actually one of the more intuitive things the brain does once you sit with it long enough, but because the implications of it are the kind that, once you've actually registered them, you can't quite unregister, which is to say every memory you have ever retrieved has been quietly altered by the act of retrieving it. Every single one. I'll come back to that. There's a window, and I want to be careful here because window makes it sound more defined than it actually is. More like a door that opens and closes cleanly, when really it's more like a period of softness, a stretch of time where a memory becomes temporarily vulnerable after a memory is retrieved, during which the physical trace of that memory can be changed. This period is called reconsolidation, which is, let me back up, because the word reconsolidation implies that consolidation happened once and is now happening again, and that's sort of right, but also slightly misleading. What's actually happening is that retrieval itself destabilizes the memory. The act of remembering something causes the synaptic connections that hold that memory to become briefly labile, which is a neuroscience word for unstable, or maybe temporarily soft is closer to how it actually behaves. And during that softness, those connections are open to modification before they restabilize. The modification is not random. What gets incorporated into the memory during this window is whatever is present in the brain's environment at the moment of retrieval. New information, updated context. The emotional state you happen to be in right now, which may be very different from the emotional state you were in when the memory originally formed. So the memory that restabilizes, the one that gets written back into the network after retrieval, is not identical to the one that was retrieved. It has been, in some measurable molecular sense, updated. The proteins involved in maintaining the synaptic connections are broken down and rebuilt, and the rebuilding incorporates the present moment, which is, I keep circling this because I'm not sure I have a clean way to say it, which is genuinely strange. The past is being edited using the present, not erased, not replaced, edited, like a document that gets slightly rewritten every time it's opened. I'm squinting at my notes right now, handwritten ones, which was probably a mistake because my handwriting after a certain hour becomes essentially decorative. And there's a study I've circled twice that I want to mention here. Researchers gave people a sedative that blocks the protein synthesis required for reconsolidation. They did this after triggering a specific fear memory, during the window when the memory was destabilized. And the emotional charge of that memory was significantly reduced in subsequent tests. The memory was still there. The factual content remained. But the restabilization had happened under conditions that prevented the full reconstruction of the emotional component. The memory came back quieter, which is, and I realize this is a slightly uncomfortable direction for a bedtime chapter to go, which is potentially useful, therapeutically, for memories that came back too loud in the first place. But the reconsolidation window is not just a therapeutic curiosity. It's running all the time, in ordinary memory, in ordinary lives, without any intervention at all. Every time you remember your first day somewhere, a school, a job, a city, the memory that restabilizes afterward has been touched by who you are now, by what you know now that you didn't know then, by how you feel about that period of your life from the distance of the present moment, and you have no awareness of this happening. The memory feels like a retrieval. It feels like accessing something that was sitting there unchanged, waiting. But the version that gets written back is subtly different from the version that was retrieved, slightly more consistent with your current understanding, slightly more coloured by your current emotional state. This is not a malfunction. It may actually be the specific biological mechanism by which experience becomes something more than a collection of recordings, by which the past gets integrated into the present rather than simply stored alongside it. The editing is what allows memory to be living rather than archival. And the thing is, this means that the very act of thinking about a memory tonight, in this quiet, in the particular emotional register of tiredness and stillness and the end of a day, is itself a form of editing. Whatever surfaces in the drift before sleep, whatever old thing gets briefly retrieved and then released, will restabilize slightly differently than it was before. Not dramatically, you won't notice. But the molecular machinery of reconsolidation doesn't require your attention or your permission. It runs on the fact of retrieval alone. The memory was accessed. The window opened. Whatever was present during that window gets incorporated. Tonight's version of the past is slightly different from yesterday's version. Tomorrow it will be different again. Not in ways that announce themselves, not in ways that feel like change from the inside. The memories feel stable because the editing is too gradual, too molecular, too far below the threshold of conscious experience to register as revision, which is, I suppose, either deeply unsettling or quietly freeing depending on the memory. The ones you'd like to update are being updated, slowly, every time you retrieve them in a different emotional context. The ones you'd like to preserve exactly as they were, those are being updated too. There's something almost circular about this whole chapter, which feels appropriate. You started tonight carrying memories that have already been edited by every previous retrieval. Whatever you retrieve tonight, deliberately or in the half-aware drift before sleep, has entered its reconsolidation window, has become briefly soft, has incorporated something of tonight's particular quiet before restabilizing. By morning, the past will be very slightly different than it was today. It always is. My tea has gone completely cold. I've been staring at a paper, printed, because I find I actually read things better on paper, even though the ink smeared slightly on the left margin, and now my index finger is faintly grey, and the paper is about REM sleep in neonates, which is, I'll admit, a topic I find genuinely strange in a way that most neuroscience doesn't manage to be strange anymore after you've spent enough time with it. Newborns spend somewhere between 50 and 80% of their sleep time in REM, 50 to 80, which is to say, the number that applies to healthy adults, the number we've been working with this entire chapter sequence, is roughly 20 to 25%. Newborns are operating at somewhere between 2 and 4 times that amount. And here is the thing that makes that number actually weird, rather than just statistically large. They have almost nothing in the form of episodic memory to consolidate. Wait, let me rephrase that, because almost nothing isn't quite right either, and I don't want to overstate it. Fetuses in the later stages of gestation are already receiving sensory input. Sounds from outside the womb, muffled, low frequency, but present. The rhythm of the mother's voice, which newborns demonstrably recognize and prefer within hours of birth, which tells you something about what was being encoded in those final weeks before the world started. There are implicit, procedural things being laid down, the beginnings of sensory maps, the very early scaffolding of a nervous system learning what it is. So it's not nothing, but it is, compared to what an adult brain is doing with 50 to 80% REM, it is a conspicuously small amount of material to be consolidating so aggressively, which raises the question, if REM sleep is primarily for memory consolidation and emotional processing, what is a 3-day-old brain doing with all of it? I have a complicated relationship with the leading hypothesis here, which I'll get to. But first, the observable stuff, because I think it's worth just sitting with before explaining it. If you've ever watched a newborn sleep, really watched, for more than a few minutes, you've probably seen it. The flickering eyelids, the small sudden facial movements that look startlingly like expressions, something that resembles a frown, then something that resembles, and this is the one that gets people, something that resembles a smile, the tiny, almost imperceptible twitches in the fingers and toes, the occasional sound somewhere between a murmur and a squeak that seems to come from nowhere. All of this is happening during REM, and the traditional interpretation, the one that gets repeated in parenting books, the one that tends to make new parents feel warmly reassured, is that the baby is dreaming, that those flickers and expressions are the surface traces of some kind of nascent inner experience. I suppose that might be true. I genuinely don't know. Nobody does, which is actually the honest answer and not a deflection. The more current hypothesis, the one that a lot of developmental neuroscientists find more compelling, though it's not settled, not even close, is that neonatal REM is less about processing experience and more about building the infrastructure that will later process experience, which is to say, the brain is using REM sleep to wire itself. The specific mechanism proposed involves the spontaneous activation of sensory and motor circuits during REM, those twitches, those flickering movements, functioning as a kind of self-generated stimulation. The sleeping newborn's brain is essentially sending test signals through its own developing circuitry, activating pathways, checking connections, strengthening the neural architecture that will eventually support coordinated movement sensory integration, the early stages of perception and response. The twitches are not random. Researchers, Mark Bloomberg's lab at the University of Iowa has done a lot of this work, and it's genuinely some of the more careful developmental neuroscience being done right now, have found that neonatal twitches during REM are highly patterned. Different body parts twitch in sequences that correspond to the developing sensory motor map in the brain. The brain is, in a measurable sense, mapping itself, using the motor output to refine the sensory feedback circuits that will eventually tell the body where it is in space. It's not dreaming, or, wait, actually, that's too definitive. It might be dreaming. The two things aren't mutually exclusive. It might be dreaming while also wiring itself. And the thing is, this reframes what REM sleep is doing across an entire lifespan in a way I find quietly significant. In adults, REM consolidates and integrates existing experience. In newborns, REM appears to be generating the very structures that experience will later flow through. The same sleep state serving what looks like a completely different function. Or maybe, if you squint at it from a certain angle, the same fundamental function at different scales, building and maintaining the brain's capacity to learn. Which leads to something that I don't think gets said enough. The amount of REM sleep an organism needs across its lifespan corresponds fairly closely to how much neural development and reorganization is happening at that moment. Newborns are enormous amounts. Young children and still significantly elevated compared to adults. Adolescence, another spike which corresponds to another massive period of neural reorganization, the pruning and reshaping of the cortex that happens across the teenage years. Adults, lower, but still essential. The older I get, the more I find that slightly humbling, which I realize is a strange thing to say, but the idea that REM sleep is, at some level, always about building or maintaining the brain's learning architecture, that it never stops being developmental, even in a 40-year-old lying in the dark, that does something to how the whole process feels. I'm looking at a photograph, not a diagram, an actual photograph taken through a specialized microscope of developing neural tissue. Rat pup, not human, but the cellular architecture is similar enough for the comparison, and the density of it. The sheer crowded complexity of axons reaching toward each other, dendrites branching and branching again, synapses forming and being pruned and forming again. All of that is happening during sleep. Not all of it. Development continues during waking hours too, obviously, but the sleep-dependent component is real and measurable, and disrupting it in developing animals has consequences that are, the consequences are significant enough that researchers take neonatal sleep disruption seriously as a developmental variable, which is a careful way of saying the sleeping newborn is doing something important. Possibly the most important neurological work of an entire lifetime, compressed into those first weeks and months when the brain is most plastic and most hungry for the activation that RM provides. The expressions on a sleeping newborn's face, the frown, the almost smile, remain unaccounted for by the wiring hypothesis alone. Maybe they're artifacts of spontaneous motor circuit activation, random outputs of a system testing its own facial musculature the same way it tests its fingers and toes. Maybe there's something else. I don't know how to end this particular thread cleanly, which is to say I don't think it ends cleanly. The honest position is that neonatal dreaming, whether it exists, what it would even consist of in a brain with so little experience to draw from, whether the concept of dreaming applies to a nervous system that hasn't yet developed the structures that support adult dreaming, is genuinely open. The cold tea is still cold. The newborn, wherever it is probably still twitching. And somewhere in that twitching, something is being built that will last a lifetime, though what exactly is being built, and how much of it involves anything resembling experience, remains one of the more quietly unresolved questions in developmental neuroscience, which I find honestly more satisfying than a clean answer would be. I've been shifting in this chair for the last 20 minutes, not because the chair is uncomfortable. It's fine, it's a perfectly adequate chair, but because there's a specific kind of restlessness that sets in when you've been awake longer than your brain was really designed for. A low-grade background friction. The sensation of thoughts that won't quite complete themselves before another one starts, which is, as it happens, not just a feeling. It's a fairly accurate description of what's actually happening neurologically when sleep pressure builds and the brain starts cutting corners on which cognitive processes it bothers to run properly. And memory is not, I want to be clear about this up front, memory is not the first thing to go. That surprises people. It surprised me when I first got deep enough into the sleep deprivation literature to see the actual sequence of degradation, because the intuitive assumption is that memory is fragile, that it's the canary in the coal mine, that a tired brain is first and foremost a forgetful brain. It's more complicated than that, and more selective, which is actually more unsettling in a way. What goes first is sustained attention, not memory, not reasoning exactly. Attention, the capacity to hold a thread of focus on a single thing for more than a few seconds without the brain quietly wandering somewhere else, and then snapping back like a rubber band that's losing its tension. This degradation begins earlier than most people realize. After roughly 16 hours of continuous wakefulness, which is to say, a completely ordinary day for a significant portion of the adult population, measurable lapses in sustained attention start appearing in cognitive testing, not dramatic lapses. Not falling asleep at the desk lapses, small ones, micro sleeps, periods of 1 to 15 seconds where the brain essentially drops into a sleep-like state without the person's awareness, the eyelids not necessarily closing, the posture not changing, but the neural activity shifting in ways that mean whatever was being processed during those seconds was not actually processed. The brain is taking unauthorized naps inside the waking day, and the person it belongs to almost never notices. Memory degradation under sleep loss is, let me try to say this carefully, because the research is genuinely more specific than sleep deprivation hurts memory. And the specificity is the interesting part. Encoding is impaired before retrieval, which is to say, a sleep-deprived brain can still access existing memories reasonably well, at least in the short term. What it struggles with is forming new ones. The hippocampal encoding process, the laying down of new traces, the initial synaptic changes that create a retrievable memory from a fresh experience. This process appears to be particularly vulnerable to sleep loss. Studies using neuroimaging show significantly reduced hippocampal activity during encoding tasks in sleep-deprived participants. The structure is there. It's just not engaging properly with incoming information. So the sleep-deprived person at the meeting, or in the lecture, or having the conversation, they're not retrieving information poorly. They're failing to store it adequately in the first place. The experience is passing through without leaving the depth of trace it would leave an arrested brain. Tomorrow, they won't remember it well. Not because recall failed, because encoding was shallow. There's a difference. It matters. And the thing is, and this is the part I find genuinely uncomfortable to sit with, the person experiencing this degraded encoding is almost always unaware of it. There's a consistent finding in sleep deprivation research that subjective assessments of cognitive performance diverge from objective performance measures as sleep loss accumulates, which is a careful way of saying people think they're doing fine when they're not. The brain, under sleep pressure, loses some of its capacity to accurately monitor its own functioning. The metacognition, that's a slightly jargony word, the self-monitoring, the ability to notice that you're performing poorly, that degrades alongside the performance itself, so you're encoding less, and you don't know you're encoding less, both at once. Now, the selectivity, because not everything degrades equally, and the pattern of what degrades faster versus slower tells you something about what the brain is prioritizing when resources are scarce. Emotional memories, as mentioned in an earlier chapter, are encoded with amygdala reinforcement that gives them a kind of metabolic head start. Under sleep deprivation, this preferential encoding appears to persist longer than neutral memory encoding. The sleep-deprived brain continues to lay down emotionally significant experiences with more fidelity than it manages for neutral ones, which means the things you remember from a sleep-deprived period tend to be disproportionately emotional. The argument, the embarrassing moment, the thing that made your chest tight, while the neutral context around those moments blurs and thins. A biased record, weighted toward the difficult. I have a mild grudge about this, honestly. The brain under stress already tends to encode negative experiences more readily than positive ones. There's an entire negativity bias literature that I won't drag us into right now, and sleep deprivation appears to tilt that bias further. You lose the ordinary parts of a hard day first. What remains is just the hard parts, encoded with the particular fidelity of emotional reinforcement, sitting cleanly in memory while everything around them has gone soft, which seems unfair. It is unfair. The biology doesn't care. Procedural memory, the kind that handles skills, motor sequences, the learned automaticity of doing something your body knows how to do, this holds up better than episodic memory under sleep loss, which is why a sleep-deprived person can still drive a car, still type, and still perform familiar physical routines with reasonable competence, even while their capacity to form new episodic memories is substantially compromised. The skills are stored differently, in different structures, the cerebellum, the basal ganglia, that appear less sensitive to the specific kind of degradation that sleep loss causes in hippocampal-dependent memory. They were learned across many repetitions. They don't require the hippocampuses to access them, so the sleep-deprived driver is not retrieving their driving skills poorly. They're just not noticing the child on the bicycle until, well, That's a different chapter and a different topic, and I don't want to go there right now. The consolidation that should be running tonight, the overnight replay, the hippocampal to cortical transfer, the M-dependent emotional processing, all of this requires sleep that is both long enough and architecturally intact. Sleep that cycles properly through its phases, a night that isn't cut short or fragmented by noise or stress, or the specific modern habit of checking a phone at 2 a.m. and then lying awake for 40 minutes with the fully activated visual cortex. One bad night does recoverable damage to the night's consolidation work. Several bad nights in a row start accumulating deficits that a single recovery night doesn't fully address. The research on this is fairly consistent and fairly uncomfortable if you think about it, while also thinking about how many consecutive nights most adults actually manage adequate sleep. I'm going to stop thinking about it. Right now, the fact that you're lying still, that the lights are off, that the day has been released or is being released, that the body is doing what it needs to do, this is the corrective. Not a dramatic one, not a complete one if recent nights have been short, but the encoding that happens during tomorrow's waking hours will be better for this night. The hippocampuses will engage more fully with whatever tomorrow brings. The traces will be deeper. I suppose that's enough for now. The chair is creaking every time I shift. I should probably just go to sleep. There is a wave moving through your brain right now, not metaphorically. An actual wave, a large, slow, synchronized oscillation of electrical activity that sweeps across the cortex in a rolling pattern like, and I want to be careful here because the comparison I'm reaching for is almost too clean, like a tide coming in across a very wide, very shallow shore, the whole surface moving together, not chaotic, coordinated in a way that if you saw it rendered visually on an EEG readout, would look almost too regular to be biological. I've stared at those readouts longer than is probably normal for someone who isn't a neuroscientist by training. There's something about the shape of them, the slow wave rendered in ink on paper or pixels on a screen, that feels almost architectural, like looking at a cross-section of something that was built other than grown. It was grown, obviously, but still. The slow oscillation, that's the technical name, which is accurate and completely characterless, occurs during the deep phases of non-REM sleep. Stage 3, if you want the clinical designation, what used to be called delta sleep, before the sleep stage classification system got revised and complicated things. The waves themselves cycle at roughly 0.5 Hz to 1 Hz. Which is to say, somewhere between one full oscillation every 2 seconds and one every second. Enormously slow by the standards of the brain's usual electrical activity. During waking hours, the cortex operates at much higher frequencies, beta waves, gamma waves, the fast flickering activity of a brain processing the present moment. The slow oscillation, by comparison, is the brain doing something that has almost no waking equivalent, a kind of electrical breathing, an inhale and an exhale at the scale of the whole cortex, repeated through the hours of deep sleep with a patience that the waking brain simply does not have the conditions to produce. Each slow oscillation has 2 phases. The first is called the up state, a period of widespread cortical activation, where neurons across broad regions of the cortex fire together in a burst of coordinated activity. This lasts roughly a second. Then comes the down state, a period of near sense where the same neurons become hyperpolarized, their electrical activity suppressed, the cortex going quiet in a way that has no real equivalent in waking neurophysiology. Silence, then activation, silence, then activation. Repeated, cycling, rolling across the brain in a slow coordinated sweep that, and this is the part I keep coming back to, the part that keeps this chapter from being just a description of electrical patterns, that appears to be the mechanism by which the hippocampus and the cortex talk to each other during sleep. Here's how it works. Or, wait, let me be more honest about that phrasing. Here's the current best understanding of how it works, which has been substantially refined over the last 15 years and will probably be refined further. The hippocampus, during slow-wave sleep, is generating its own faster oscillations. The sharp-wave ripples mentioned in an earlier chapter, those brief high-frequency bursts that appear to represent the compressed replay of daytime experiences. These ripples occur preferentially during the upstate of the slow oscillation, which is to say the hippocampus times its replays to coincide with the moments when the cortex is most activated, most receptive, most electrically ready to receive incoming information. And nested within the upstate, alongside the sharp wave ripples, is another oscillation. Sleep spindles, bursts of activity at around 12 to 15 Hz that last for roughly half a second to 2 seconds, which appear to play a role in actually transferring the information from hippocampal replay into cortical storage. The spindle is, in some functional sense, the bridge, the moment of handoff. So the full picture, during a single slow oscillation, looks something like this. The cortex activates in the upstate, a hippocampal sharp-wave ripple fires carrying a compressed memory replay. A sleep spindle bridges the two structures. The cortex receives the signal and begins the slow process of incorporating it. And then the downstate arrives and everything goes quiet again. 2 seconds, maybe less. And then it repeats. I find sleep spindles, I should say this openly, I find sleep spindles slightly underappreciated. They get mentioned as supporting players in the slow oscillation tree, which they are. But the research on individual differences in spindle density is genuinely interesting and doesn't get nearly the attention it deserves outside of specialized literature. People vary substantially in how many sleep spindles they generate per night. And this variation correlates, not perfectly, not deterministically, but consistently enough to be meaningful, with performance on memory consolidation tasks. More spindles, better overnight consolidation. The correlation holds across studies, across different memory types, across different age groups, which is to say, some people's sleeping brains are running this hippocampal to cortical transfer more efficiently than others, and the difference is at least partly visible in the density of those half-second bursts of 12 to 15 Hz activity that most people have never heard of. The brain's overnight filing system runs at different speeds in different people. Nobody knows exactly why. There's a quality of the slow wave that I want to try to describe, because I think the electrical description alone doesn't quite capture what it actually means for the brain as a whole. During the downstate, the period of near silence, the hyperpolarized quiet between activations, the cortex is not merely resting. It is, in a specific neurophysiological sense, being reset. The neurons are recovering their electrochemical gradients. The synaptic resources that were used during waking are being replenished. The metabolic cost of the previous upstate is being cleared in preparation for the next one. The wave is not just communication, it is maintenance. Each cycle cleans something and builds something and transfers something in a sequence so fast and so repetitive that across a single night of adequate sleep it runs hundreds or thousands of times, the exact number depending on sleep duration, sleep quality, age, and a handful of other variables that the research is still characterizing. Hundreds of times, each one carrying a fragment of today towards something more permanent. I'm sitting here. The room has gotten colder in the last hour, the particular cold of a night that's moved past midnight into the part of the night that doesn't have a good name. And I'm thinking about the fact that this wave, right now, is running through the brain of anyone listening to this who has already fallen asleep, which is, I hope, most of you. The slow oscillation doesn't care whether you know about it. It doesn't require your attention or your appreciation. It runs on the conditions of sleep alone, on the neurochemical state that deep sleep creates, on the reduced input from the outside world, on the particular electrical environment that only the sleeping brain generates. And in that running, in that quiet repetitive cycling between activation and silence, something from today is being moved. Not all of it, not tonight, but some of it, the pieces that were encoded most clearly, that carried the most signal, that the hippocampus has been holding in careful temporary custody since they formed. Some of those pieces are making their slow way toward the cortex, carried on the back of a wave you'll never feel, but that is right now doing some of the most important work your brain will do all week. The wave moves, you don't notice. Both of those things are, I think, exactly right. It was there. You know it was there, not as a belief, not as an assumption, but as a felt certainty in the first 2 or 3 seconds after waking. Something was happening. Something with a location, a feeling, maybe a person or a colour or a specific texture of wrongness that dreams sometimes have. Something that was, in those first seconds, still present enough that you could almost reach back into it. And then it was gone. Not faded. Gone. The way a word disappears when you reach too hard for it. The reaching itself seems to collapse the thing you are reaching for. You felt this. Probably this morning, probably hundreds of mornings. I've spent an embarrassing amount of time trying to catch dreams on the way out, lying completely still after waking, not moving a single muscle, eyes still closed, attempting to hold whatever was there by not disturbing the air around it. Sometimes it works for another few seconds. Sometimes the dream extends just long enough to add one more image before it goes, but it always goes. And the reason it goes, which took me longer to find a satisfying answer to than I expected, is not mysterious once you understand what the dream was made of in the first place. Dreams are not stored memories that get played back during sleep. I want to say that clearly because the intuitive model most people carry, the one where dreams are a kind of cinema, where the brain is screening content that it has filed away somewhere accessible, that model is wrong in ways that matter for understanding why dreams disappear. Dreams are generated in real time from the activity of a brain running in a specific neurochemical state, which is to say, the dream is not retrieved when you dream it. It is constructed, moment to moment, from the associative firing of a cortex that has lost its norepinephrine, that has lost the filtering that normally constrains which neural connections are allowed to complete themselves. The dream is the output of that unconstrained state, not a recording, a live event. And live events, when the conditions that generated them change, don't leave recordings behind, they just stop. When you wake, or even when you transition between sleep stages, which happens multiple times a night in ways you don't register, norepinephrine returns, not slowly, fairly quickly as neurotransmitter changes go. The locus coeruleus, which went nearly silent during REM, begins firing again. The filtering resumes, the associative looseness that allowed the dream's logic to hold together, the corridor that stretched, the person who was two people, the building that was somehow also a childhood memory, that looseness no longer has the chemical support it needed. The dream's internal coherence collapses. Not because it was never coherent, it was in the particular way that REM state neural activity is coherent, which is its own kind of logic, but because the brain reading the dream upon waking is a chemically different brain than the one that generated it. And the waking brain, applying its reinstated filtering to the fragments it can still access, finds that they don't hold up. The connections don't survive scrutiny, so they dissolve. What remains, if anything remains, is almost always the most recent fragment. The last image, the final emotional register, the ending without the middle or the beginning, sometimes not even that. There's a theory, which I find plausible but want to be careful about overstating because the evidence is suggestive rather than conclusive. that the act of waking too abruptly, particularly from deep REM, gives the norepinephrine return less time to be gradual, which means the chemical environment shifts faster, which means even less of the dream survives into the waking state. Whereas waking slowly, naturally, without an alarm, drifting up through lighter sleep stages before full waking, gives a slightly longer transition window where some of the dream's content might be consolidated into a form the waking brain can hold, which would explain why dreams remembered with most vividness tend to be the ones from slow, natural wakings, and dreams from alarm clock mornings tend to leave nothing at all. I have a mild but persistent resentment toward alarm clocks on behalf of this researcher. Not a reasonable resentment. Alarm clocks are necessary. But the specific violence of a loud alarm cutting through REM sleep, the immediate 0 to 60 norepinephrine spike that must accompany that kind of startled waking, feels, now that I understand the mechanism, particularly hostile to whatever the dreaming brain was in the middle of doing. The dream doesn't just dissolve, it gets interrupted mid-construction. There's another layer to this that I find slightly strange every time I return to it. Dreams that are remembered, the ones that survive into waking long enough to be encoded as episodic memories, become in that encoding something fundamentally different from what they were during sleep. They become hippocampal-dependent memories, subject to all the same consolidation processes, the same reconsolidation vulnerability, the same gradual revision across subsequent retrievals as any other memory. Which is to say, the dream you remember from 3 years ago is not the dream you had. It is a memory of a memory, a hippocampal encoding of whatever fragment survived the waking transition, consolidated across the nights that followed, revised each time it was retrieved, shaped by everything you have thought about it since. The dream has been edited into something the waking brain could hold. What the sleeping brain actually generated is gone. This leaves me, and I don't think I can resolve this cleanly, so I'll just say it. This leaves me genuinely uncertain about what dreams are, in any meaningful sense. Not scientifically uncertain. The neuroscience of dream generation is fairly well characterized at this point, but uncertain about what they mean, in the ordinary human sense of the word mean. Whether the thing we call a dream, the thing we sometimes tell other people about over breakfast, the thing that occasionally feels significant in ways we can't quite articulate, whether that thing bears enough resemblance to the original REM state neural event to carry any of its content faithfully, I'm not sure it does, which is either a deflating thought or a liberating one. I keep going back and forth. The corridor from the dream I mentioned earlier in this script, the one that kept stretching, I've thought about it a few more times tonight, while writing other chapters, and each time I've retrieved it, I've probably altered it slightly, added a quality it didn't have, smoothed something, lost a detail that was there in the original fragment. The corridor is being edited. By the time this script is finished, the dream it came from will be further from whatever actually happened in that sleeping brain than it was when I first mentioned it, and the sleeping brain that generated it has no record of it at all. The dream is only in the waking. It was only ever in the waking. Whatever it was during sleep is already somewhere else entirely, dissolved back into the chemistry that made it, which has since moved on to other work, leaving no trace and no forwarding address. There's a moment, you probably won't catch it, almost nobody does, just before the alarm goes off. A kind of surfacing, a shift in the quality of sleep that isn't quite waking, but isn't quite sleeping either. The body repositioning slightly, a thought that's almost a thought. The sense of the room arriving before the room has technically arrived. And then the alarm, or the light, or just morning with no clear announcement. But the thing is, that surfacing wasn't random. I've been sitting with this chapter for a while tonight. Longer than the others, actually. Not because the biology is more complicated, but because there's something about it that keeps snagging my attention in a way I find hard to move past quickly. The body was expecting morning, not in a vague general sense, in a specific chemical anticipatory sense. The cortisol was already rising. The core body temperature was already beginning its slow climb back upward from the lowest point of the night, which bottomed out somewhere around 2 or 3 in the morning while you were completely unaware of it happening. The melatonin, which had been elevated since sometime in the evening, was already declining. All of this was in motion before the first photon of morning light touched your retina. The body was not waiting to be told. The system responsible for this is called the circadian clock, which is one of those scientific names that sounds more metaphorical than it actually is, because it really is a clock, or close enough to one that the distinction barely matters for this conversation. It runs on a cycle of roughly 24 hours, driven by a cluster of about 20,000 neurons in the hypothalamus called the suprachiasmatic nucleus. The SCN, if you want the abbreviation, which I'll use because typing suprachiasmatic nucleus repeatedly at this hour feels slightly aggressive. The SCN receives direct light input from the retina via a dedicated pathway, not the visual pathway, a separate one, running specifically to deliver light timing information rather than visual information. This is how the clock stays synchronized to the actual day-night cycle, rather than drifting according to its own internal rhythm, which runs slightly longer than 24 hours in most people and would, without daily light correction, cause the sleep-wake cycle to gradually shift later and later over time. Light resets it. Every morning, without fail, a small molecular correction that keeps the internal clock aligned with the external world. But the anticipation, the rising cortisol, the climbing temperature, the pre-dawn preparation, this happens before the light correction, not because of it. Which is to say, the SCN is not responding to morning, it is predicting it. The clock, calibrated by yesterday's light exposure and the days before that, has calculated approximately when morning should arrive and has begun preparing the body for waking somewhere in the hour or two that precede it. Cortisol, released from the adrenal glands in response to signals from the SCN, starts rising roughly an hour before typical wake time. Not to stress levels, to something more like a gentle readiness, a slow biochemical preparation for the transition from sleep to waking that the body apparently prefers not to make abruptly. The cortisol morning rise, it's sometimes called the cortisol awakening response, though that name applies more specifically to the spike that occurs in the first 30 minutes after actual waking. This pre-dawn preparation is one of the more quietly elegant things the body does, in my opinion. Is the body being considerate of itself, getting things ready before they're needed. Core body temperature follows a similar anticipatory curve. During deep sleep, body temperature drops not dramatically by about 1 to 2 degrees Celsius, but consistently and measurably, and this drop is itself part of what facilitates deep sleep. The cooler core temperature supports the neurochemical conditions of slow, low-wave sleep in ways that researchers are still fully characterizing, which is why a cool room helps people sleep better, and why a hot bath before bed can paradoxically improve sleep onset. The rapid cooling afterward mimics the body's own pre-sleep temperature drop, nudging the system in the direction it was already trying to go. And the thing is, I keep coming back to the timing of all of this because it keeps surprising me no matter how many times I read it. The temperature starts rising again before the alarm, before the light, before any external signal has been received. The clock is running the show. The external world is just confirming what the internal world already calculated. Melatonin, which most people know as a sleep supplement, is actually more precisely a darkness signal than a sleep signal, which is a distinction that matters more than most people realize. The pineal gland releases it in response to darkness, and it rises across the evening in a way that communicates to the body's systems that night is happening, and that the appropriate biological responses to night, including the initiation of sleep, should begin. By the pre-dawn hours, the darkness signal is already winding down, not because light has arrived, because the clock knows light is coming. Melatonin levels begin declining in the second half of the night, pulled down by the same anticipatory clock signaling that is simultaneously raising cortisol and temperature. The body is, in a chemical sense, already beginning to leave the night before the night is over. I find this almost unreasonably interesting, which I realise is a strange thing to say. It's not the most dramatic biology covered in this script. It doesn't involve the structural complexity of hippocampal replay or the neurochemical intricacy of REM sleep. It's just a clock, running, predicting, preparing. But there's something about the image of a body lying in complete stillness and apparent unconsciousness, while inside it a 20,000 neuron cluster in the hypothalamus is quietly coordinating a cascade of hormonal and thermal adjustments in preparation for a morning it has never technically seen yet. Something about that image that I find disproportionately moving. The body is always already working, not just maintaining, anticipating, leaning forward into time. There's also something worth saying about what disrupts this system. The circadian clock is calibrated by light, and it is recalibrated every day by the light exposure of that morning. When light exposure is inconsistent, when some mornings are bright and some are not, when artificial light at night delays the evening melatonin rise, when the sleep schedule shifts significantly between weekdays and weekends, the clock's predictions become less accurate. The pre-dawn cortisol rise doesn't align properly with actual wake time. The temperature nadir shifts. The body begins a kind of internal jet lag, technically called social jet lag in the research literature, where the biological clock and the social schedule are running at slightly different times, and the body is perpetually preparing for a morning that arrives either too early or too late relative to what the clock calculated. The preparation still happens. It's just aimed at the wrong time. Right now, at whatever hour this is for you in whatever room, with whatever particular quality of darkness, the SCN is running. It has been running since before you fell asleep. It ran through every sleep stage, every cycle, every dream you had and lost. It noted the timing of the night. It has been updating its prediction of when morning should arrive, and it has already or probably begun the slow pre-dawn cascade. The cortisol is moving, the temperature is turning, the melatonin is on its way down. You won't feel any of this. You'll just wake up, and it will feel like waking up because morning came. But the morning the body is waking into is one it already knew was coming, prepared for, aimed itself toward, through an unbroken chain of chemical signaling that ran underneath everything else this entire night. The clock doesn't stop, it doesn't need to be told, it just keeps going quietly, accurately in the dark. And that, I think, is where this particular journey ends, not with a dramatic conclusion, but with something small and true. Your body ran all night. It cleaned itself and transferred memories and processed emotions and predicted the morning and regulated its own temperature. And it did all of this while you were completely unconscious and mostly unaware, and probably dreaming about a corridor that kept stretching. It did all of this for you, without asking. If you made it to this point, if this script carried you through some part of a night or helped the day release a little more easily, that means something to me. It genuinely does. The sleepy biologist exists because people keep showing up for it, keep listening, keep sending the occasional message that says, "I fell asleep during chapter 4, and I mean that as a compliment." Those messages keep this going more than anything else. If you'd like to keep this space alive, there's a membership option, quietly sitting there if you ever feel like it. And if a super thanks ever feels right after a particularly good night's sleep, those land in a way that's hard to overstate. But mostly, if you like this, telling one other person about it is worth more than almost anything else you could do. Word of mouth is how small channels survive. Leave a comment if something in tonight's video lodged somewhere unexpected. I read them, all of them, usually late at night, in the same chair I've been sitting in all evening, with tea that has gone cold again. Subscribe if you want more nights like this one, and if you're still awake, you should probably fix that.